So I wasn't, needless to say, I wasn't particularly predisposed necessarily to work on, I'm working on carrot genetics now. And of course human cytogenetics is very different. But so I didn't have a particular idea of what I wanted to be doing, and I ended up taking this job sort of as more of a flip of the coin than anything else. So what I do with what I've been doing with carrot is basically establishing a genetic foundation for the crop. We're looking ultimately at improving the crop, and in a minute I'll say what that means, and you can tell me what it means too. But that's the goal of the project, is to improve carrots, and since I'm a geneticist, improve them from a genetic standpoint. One thing we're doing is looking for genes for disease resistance that will help the growers grow the crop with no or at least less pesticides. So we're looking at organic production too as an alternative or an additional way to grow carrots. So I had an undergrad this semester look at some genes in the pathway that result in the accumulation of purple pigments in carrots. There are purple rooted carrots. So now we know some genes in that biosynthetic pathway, and we know there's two genes involved in purple carrots versus non-purple, and the question is, are those genes in that pathway, and if so, which genes are they? Well, you can go through a number of genetic and biochemical evaluations and answer that question. So anyway, we do quite a bit of work on biochemical genetics of these compounds as well to get some ideas on what's going on there. It also is important from this whole question of carrot domestication, because wild carrots are never orange, they're always white rooted. And so the question is, where does that gene come from? And what can it tell us about the history of the crop? So, anyway, so that's the kind of range of stuff that we do. If there's a new disease for carrots, then what we'll do is get samples of all these seeds, expose them to the disease, and see if we can find some genetic resistance. And that's been a very effective tool for improving pretty much all the crops that you eat. Something like potatoes, there's almost no potatoes that you eat that don't have wild relatives of potatoes that were intercrossed with cultivated potatoes by breeders in the last 100 years to bring in resistance to viruses and diseases, to improve storage ability, things like that. So these carrots were grown we planted the seed in October in Southern California, about 8 miles north of the Mexican border on a research station there. We harvested these carrots in March and shipped them back to Madison from Southern California. So this is an example of evaluating a trait so that we then can start at this level and ask the question, how many genes are involved in controlling root color? And it looks to me like probably one gene or a little bit. You expect roughly a 3-to-1 segregation based on other things we know. Virtually every consumer would prefer to have sweeter carrots. Very few people would dispute that. So one thing we're looking at is breeding for sugar, and I do actually flavor evaluate thousands of carrots every year. It's not as sexy as wine tasting, but I do it. But the other thing we do is we actually analyze sugar content and push it through a stainless steel column that is, it's running right now so I can't open it, but it's a stainless steel column that looks like this. The sample goes in here and it's a mixture of sugars or carotenes or anthocyanins, and then as it goes through the column, the packing that's in this column separates it so that when at the other end of the column, what started out as a mixture of these different pigments or sugars is separated into individual sugars so we can quantify each one separately. So what I've been collecting, I think this is about my 10th or 12th collecting trip. I've been collecting primarily in the Mediterranean to Central Asian part of the world, quite quite a few times, 10 or 12 times now, with the purpose of picking up wild relatives of carrot and to some extent wild relatives of garlic, with this idea that more diversity is a good thing to have on hand. In some cases, we pick up wild relatives that are becoming endangered because as people spread out, they tend to put parking lots where wild plants used to be growing. That's how we use this food in your crop composition data to help us understand the biochemistry and make decisions for growers and processors and consumers as we go along. So that's the kind of stuff we do, and we